Thank you, Dr. Levinson, for your wonderful introductions. I told Dr. Levinson that this will be the first time that I will not talk about DNA methylation and clinical diagnosis. Okay, because each time when I give lecture for the past 20 years, I always talk about DNA methylation. And this is the beginning of the first time I will talk about something completely different. And all the work basically just happened about 2 years ago, okay, when we moved to San Antonio, Texas. So today I would like to talk about single cell analysis. And I hope for the next 40 minutes or so to convince you that single cell analysis is important for cancer detection and prognosis as well. Okay, let me begin using prostate cancer as an example. All right, for diagnosis of prostate cancer, of course, a man, up to 50 to 60 that has PSA 4.0, and in conjunction with the positive digital rectal exam, of course, then the next recommendation likely is a needle biopsy. And we know a lot of time PSA tests produce a lot of false positive because inflammations, okay, or in prostate and urinary tract, as well as benign prostatic hyperplasia, also has a high PSA. So only 25% of men who undergo a prostate biopsy actually has a diagnosis of prostate cancer. So there's about 1.5 million of men who have high PSA positive, DRE, and has to go through that procedure, and yet 75% will be basically considered to be a normal. Now, a man may have a low PSA level, and lo and behold, 15% of men who has low PSA, and when you do the biopsy, and it produces a positive result. Okay, so one of the dilemma is also prostate cancer overdiagnosis. For example, lots of needle biopsy and overtreatment in low-grade prostate cancer. Basically, it will not basically disseminate later. Now, there's another problem of underdiagnosis, because a lot of time when a man has a Gleason score 3+3, that's considered to be low-grade, at the time of a needle biopsy, and you later find out when the patient elect to have a prostatectomy, the Gleason score basically been upgraded. Sometimes could be GS 4+3 and maybe 4+4. And roughly 30% of patients with low-grade needle biopsy had that problem. So this is become a situation is, can we have a better diagnosis? And I'm a big fan on this. Basically using exfoliated cell for diagnosis. All right, so as you can see, this is the tubular lumen, and the reason is a tumor cell easily to be shedded is due to increased activity of ECM, or extracellular matrix degradation enzyme, that contribute to the loosening of cell. Okay, so they now can be free-floating cell, or sometimes they can disseminate into the blood vessel. What I have not shown here is the also free nucleic acid, okay, which later we'll talk about that. So, since I'm a chair, one day I was in molecular medicine. I am so compelled to tell my dean, perhaps I'd like to change my department as Department of Molecular Waste Management. Reason is, you can isolate exfoliated cell from sputum, stool, urine, and ejaculate, ductal lavage, of course, and tampons. A lot of time, the needle biopsy to be discarded that we can collect, because if we want to look at exfoliated cell, that will be resources, and we don't need a lot. I like to convince you to do this type of analysis, you don't need a lot. So once we have exfoliated cell like being collected, we can do single cell targeted sequencing, and you can do now single cell omic to do transcriptomes and methylome, and also the whole genome mutation scanning. One can also do immunostaining at the single cell level using this, and of course, fish analysis to look at copy number variation of a particular site. And today I'd like to focus basically on expression analysis. We have some data on single-cell methylation analysis, but I think it's not ready to show it here. And we'll talk about some physical property of single cell. All right, so my talk will be used basically urine as an example to look at the exfoliated cell in urine, and that's come from prostate origin. So doing urine diagnosis of prostate cancer, nothing new, right? You have high PSA positive digital, and sometimes needle biopsy turned out negative, and the doctor may recommend that you do a PCA3 test. So it's a non-coding RNA gene that well known. Actually, it's been discovered, I think, by a scientist in Johns Hopkins, just in Baltimore here. Okay, so you have a digital rectal examination to stimulate, and then after that you collect the urine. And then you basically centrifuge the tube, and you collect urine sediment. Then you then isolate RNA, okay, and then do a PCR base. So right now is you have PCA3 and then a PSA level as combined together. So if you look at this chart, normal has about this range. But in patients with prostate cancer, where you can detect a lot of PCA3 floating around in the urine, this is the level. Now, if you compare here, there's actually a chunk of patients that could potentially be misdiagnosis. And there's a PCR assay basically has a sensitivity of 60 to 70% and specificity of 70 to 80 for prostate cancer diagnosis, for a patient that basically has negative needle biopsy. So the question is why we're interested in isolate exfoliate prostate cell in urine, and what is the purpose of that? And we now know cancer can be considered as a complex disease. And you can start with a normal cell, and then over the development and the collections through a conal expansion, there's basically different stage, and perhaps every cancer cell may have a different genome. Okay, so that is a highly complex situation. But this kind of intraphenotypic heterogeneity perhaps can be used to decipher conal conditions, and then take that important information to inform clinicians for appropriate diagnosis and treatment as well. So we have in the laboratory developed two platforms for non-invasive cancer detection using urinary exfoliated cell. The first platform is a depth array, where you can take a liquid that contains exfoliated cell with a sputum after appropriate treatment and put it on the chip. And so this depth array allows you to stain roughly 5 different colors, including Debbie. So here is a color. And basically, it's a chip that contains roughly 20,000 microgrid. Live cell has electrical charge. So once a live cell been positioned in an electromagnetic field, it will basically position in a specific grid. Okay, and then one you can use a different fluorescent color to pick out the property of that cell. All right, and then after that you can collect the cell and move the cell in a travel, minus plus. You can basically move the cell of interest to a parking station, and from there you can collect that for a downstream molecular analysis. So this is sort of a high throughput platform. And so one day we were doing the experiment, and someone visited us from in vitro fertilization, say, well, this is a fluorescent microscope, and you can see a specific cell that lights up, they say, a red color. How about just using a micromanipulator to pick it up? So indeed, we are able to do that, and actually it's not that difficult. Once you have the cell and then a trained technician with a 2-day training, you basically can collect single cell from all these resources here, all right? Now we'll talk about biobanking. Here I'd like to throw a wild idea. How about single cell banking? Okay, by looking at individual cells, for instance, intensity and property, and then one can bank millions of cells. I think it won't take a lot of space. Okay, we have now banked roughly 5,000 cells, from our own initiative, and perhaps that I can convince you, that Dr. Levinson, or other scientists here, that this may be another additional avenue one can do. All right, and once we have the single cell banking, then we can do single cell analysis. And this is one of the examples using microfluidic. So there's roughly 2,500 individual PCR chamber here, all congregate in a roughly 2.5 times 2.5 centimeter chip. And this is individual PCR well, okay? So we take that array and just to count every cell, okay? Live cell from a 25 cc urine of a patient with prostate cancer. And we know urine contains a mixed bag of cells. It can come from kidney, bladder, or other hematopoietic cell as well, been basically present in serum. We took PSA and PSMA as our color. Okay, here in the depth array, when a live cell that is still alive, that can be deposited in a microgrid here, shown here. Okay, one can view there's a PSA positive, PSMA positive, and DEPI. Okay, that's a cell. That's not a debris, because a lot of time when you look at urine sediment, you basically find there's a lot of debris there. Okay, so this is a PSA, PSMA negative cell, and likely come from bladder or kidney or other resources as well. So we survey, count every cell, live cell in the microgrid. For example, this patient had 549 urinary cell, with that 44, roughly 8%, that been basically identified as prostate-specific cell based on PSA and PSMA staining. All right, so you look at the PSA intensity, and I could tell you, even in a single human being, a single male, here's exfoliated cell, the expression level is basically quite different. This is a normal level. Okay, we have done the control. Men without known history of prostate cancer basically has a very modest or low level of PSA. But this patient yet, you can see roughly half has a higher PSA level. Now compare patient 14 with prostate cancer, 13% of urinary cell basically has a prostate origin. And this patient, you can see obviously have two groups of cells. One group have low PSA, and the other group has high PSA cell. Okay, this patient, patient 24, has a gradient, and patient 25. So if you think about in the good old day, when you measure a person, the PSA, serum PSA, you basically, it's a whole cell approach. This is PSA level. This patient had either greater than 4.0 or below, whatever. But when you begin to look at single cell now, you can see how heterogeneous it can be. All right, so that's why performing analysis of single cell, it's important. So we get back to that what we call cherry picking approach. All right, either a man with DRE or without DRE, and then we collect urine sediment. After that, digest a little bit with collagen so that you can briefly dissociate the clump a little bit, not too much. And then stand with PSA and PSMA. So just imagine you are in a swimming pool. You're looking at a tennis ball. That's a red tennis ball. That's a technician or a trained scientist look at under a microscope or inverted microscope. Okay, just think about that. He's looking at a swimming pool to see whether there's any red ball there. Once he finds a red ball, then the micro tip will go in. So this is a prostate cell in urine sediment. Okay, there you can see there's a lot of debris here. There's also other cell. So we've been trained in such a way, the tip, you can move the neighboring cell away. Okay, you can move the tip up and down up to a point that you think you can just literally take up that specific prostate cell and then deposit in the tube. Is this a difficult technique? I trained my wife to do that, and she's a non-scientist, but right now she's able to pick single cell. And with experience, you can pick 30 prostate-specific cell in an hour. Okay, so it's not a difficult and labor-intensive technology. On average, that we are basically survey 30 to 50 prostate cell per 25 cc urine sample, and then after that we do microfluidic PCR analysis. Okay, so when I first time showed this slide, not many people understand this. This is not heat map. Okay, this is every red line indicate the positions of the expression level of that particular cell. Okay, expression value of a single prostate cell. For example, there's 5 control here, and there are roughly 40 single line here. This is expression level based on minus delta CT after normalization with a housekeeping gene UBB1. Okay, this is second patient. This is third, a total of 5 control, sorry. And then a patient with enlarged prostate or high-grade intraepithelial prostate epithelia. Now, if you look at patient with prostate cancer, remember PCA3 has been used for routine diagnosis now, patient with prostate cancer. But if you look at now single cell, all right, there's actually a wide spectrum of expression even in patient with prostate cancer here. But when you begin to see a lot of single cell actually has a higher PCA3 expression level shown here, all right? And some patients, since we lump BPH and high-grade pin together, compared to normal, you can see it's highly heterogeneous. But within that realm of heterogeneity, that you also see high expression, some of the cell that has high expressions of PCA3. All this right now, total of 1,220 single cell that we analyze. Now, of course, in the good old days, you basically lump everything. This is control, this is volume plot. Somehow there's kind of 2 population of cell, and BPH has higher average level. And of course, we are able to confirm, indeed, patient diagnosed with prostate cancer has high PCA3 level. But even with that, you can see there's still a wide spectrum of distribution in terms of PCA3 at the single cell level. Now, how about other type of genes? What's the expression level in this 1,220 cell that we analyze? There's basically wide spectrum of value, BIK, CDK, and 1A. Look at that. To me, when I look at that, this is chaotic. The expression level, none of the single cell have the same expression value, all right? And this is another gene. It's more uniform, and E2F1. Look at the PSA. The alias of gene is KLK3, okay? And indeed, in patient with BPH, high-grade pins, and prostate cancer, and overall, has a greater PSA value than normal. And this is a keratin number 8. During this type of analysis, we find a very interesting thing, that we began to see that some of the genes, especially in prostate cancer, basically can be divided into 2 groups. Each time when I look at this data, sometimes I got dizzy. Okay, and then I have a discussion with Dr. Levinson. You have to keep things simple, and whether there's any way that one can do. And there's 6 gene here, CXCL6. It seems to me there's a bimodal distribution, and TGFBR2 as well, okay? GSK3B, frequently is upregulated in prostate cancer patients' cell. CDKN1C is non-tumor suppressor, but there's a 2 group of expression level that we can perhaps somehow simplify. And indeed, when you throw in statistic analysis, they fit into a so-called bimodal distribution, all right? So then we come out an idea. Can we now, to piece all this gene together, all right, here's a bimodal distribution, and that particular cell from a man's urinary cell, the expression level is home in here. Now, can you connect the expression level of the same cell from CSCL6 to TGF-beta? So you can do the connections. So this is a specific computational algorithm, what we call parallel coordinate plot. Okay, you can connect two or multiple genes in the same cell. All right, so this is the blonde line. It's a total of 1,200 single cell that we can connect. And lo and behold, when you look at the gene that has bimodal distribution, one can see there's basically some are up, low, low, up, or low, low. All right, and we think about perhaps that we can take the midpoint and to simplify it as 0 or 1. Okay, like for example, there's the cell that has low value of CSCL6 but high value of TGF-br2. Then perhaps we can code it at 0, 1. Okay, another cell has low value of CSCL6 and TGF-br2, then we code it as 0, 0. So this is the traveling of that single cell. Okay, high value or high low value. All right, so we began to simplify this highly complex expression pattern. Just give you an example with the now connect 3 genes for the same cell. Okay, this is 3 genes, CDKN1C, GATA3, and EIF4EBP1. So one cell has the binary code of 0, 0, 0. The other cell has binary code 0, 1, 0. So one can basically, you have 2 to 3 type. That's basically a binary code. And we can search the binary code, and lo and behold, we can travel with that single cell and establish binary code. So now, let's look at the urine sample that we collect. So with 6 genes that has a bimodal distribution, and you do parallel coordinate plot and begin to look at individual cell and their expression pattern and travel with that. They look at 3 individuals that has no history of prostate cancer, kind of highly heterogeneous. BPH, this patient and this patient, look at CSCL6 and TGF-br2, somehow the high expression value, but there's still some crisscross. Look at high-grade pin. You can begin to see a lot of cell has basically low expression value. This is patient with prostate cancer. Some of patients basically are quite obvious. When you look at the travel pattern using parallel coordinate plot, they are quite different. So we have the idea again is just to convert expression value and to binary code. Okay, this is, I don't know whether this idea is too wide or not. Let's see, let's follow pattern of a single cell. This is the expression value of CLC6. This is again the expression value of the same cell. Okay, same cell and same cell, and then you can see low, 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 high, low, low. You can basically convert that to 0, 0, 1, 0, 0. Remember, we analyze expression pattern on average, roughly 30 to 40 cells of that single patient. Okay, so this is the summation of total pattern on the background of that 1,220 cells. So this patient, we analyze a total of 32 urinary prostate cells. Okay, and there's basically has 32 lines there. Okay, and very interesting thing, you think about 2, 6 should have 64 code. If this is a random drawing, okay, random drawing is supposed to have 64 code, but of course we have only 32 cells analyzed. But again, the binary code are not random. All right, so like here we have Huang pops up. Okay, 0, 0, 0, 0, 0, 0. Okay, you can see this is the group of cells that has a binary code 0, 0, 0, 0. Some cell has 0, 0, 0, 0, 1, 0. Another different type of binary code. And altogether, we find in that urine specimen, out of 32 cells analyzed, we find there's 1, 2, 3, 4, 5, 6 chronopopulation that we can detect. Now you think about the binary code, to have two cells has that exact binary code, it's 1 out of 64 times 1 out of 64. The probability is almost 0.000 something to have a similar binary code, and yet they have the similar code 0000 here. Okay, so now it's a lot of statistics, a lot of bioinformatics, and we've been talking to our group of bioinformaticians. They come out, how about let's do polar plot analysis. Okay, take, throw in all the binary code of this 1,220 cells. And then you can draw, let's say, 200 cells, okay? And see in this random sampling, and then compare your cancer population that you analyze, all right? So this is the average value and standard deviation. This is the average value, and this is standard deviations of random sample. Okay, where their code fall into that. Now, if you compare this binary code 111101, which is very common in urine of patients with prostate cancer, it's actually 4 out of that range. So this can be called as a binary code specific for prostate cancer. All right. So altogether, you can see there's 11111, 111110, 111101, or 111100. Okay, the one coded in red, basically, based on this polar plot analysis, identify as unique binary code associated with prostate cancer. We can also identify binary code associated with high-grade prostate intraepithelial hyperplasia. The most common one is 0000. That means the 6th gene that we identify basically has a very low expression value, okay? And there's about a total of 6 codes that we can identify. How about enlarged prostate? There's also some code that fall into a unique pattern, okay, like that. And we also find there's a common code that can specify patients, an individual without prostate cancer, and this is the most common code that fall out of the standard deviation. This is 100010, okay? So the goal is to simplify complex expression pattern into a recognizable code. So we are able to, out of a total of 64 codes, identify a specific code for a group. This is group likely normal, enlarged prostate, and high-grade PINS, and also prostate cancer. Now, this is actually easy. Well, you can validate all these genes at 0. The 6th gene that we pick up actually are tumor suppressor genes. And we know tumor suppressor gene, that's in cancer that are supposed to be repressed. The gene can be silenced, no expression, and therefore you have very low expression value. And in that case, 0, we call it a low expression value. And all these are tumor suppressor gene, likely all silent, except sometimes occasionally you see there's one gene pops up. Now, why in prostate cancer this tumor suppressor become activated? And there's now increasing evidence. A lot of the so-called tumor suppressor gene can be reactivated. For example, TGF-br2 that I just give you an example is a non-tumor suppressor, no doubt about it. Okay. Now, in advanced cancer, this gene actually can be reactivated. So, tumor suppressor means suppressed, then cancer have a proliferative potential. Now, if this gene been reactivated in advanced cancer, it actually contribute to increased migration and increased cancer metastases. So when patient with this unique code, actually likely it can be predict a poor prognosis. Now let me give you an example now. Of course, this is a very complicated gene tracing plot. Okay, we perform this kind of plot and look at their crisscross and traffic pattern. It's a little bit chaotic. And then we convert to binary code. and identify whether there's any clonal populations. Our overarching goal is if this can become a diagnostic tool. This is the tool, the picture we're going to show to patients. In this case, this is a single cell that we analyze. However, in here we see there's a total of 8 clonal population in that specific urine sample. Okay, and green indicate that's a normal sample. This is also some binary call here. Okay, this is a patient. It's a normal individual with highly diverse populations. And this normal individual has 4 clonal population here. Okay? Now, let me move to a hyperplasia conditions. A major clonal population can be detected in his urine sample that has a binary code of 110001. In addition, there are 2 minor BPH clonal population can also be detected. Dr. Levinson said this kind of color-coded clonal population may not be good for some male who has colorblind. So we probably have to choose an appropriate color, all right? Now, this is a 65 years old man with PSA 4.8, a little bit high, okay, and biopsy confirmed that has a precursor lesion, no cancer. But we know high-grade pin, a lot of time it's a precancer condition. And look at that. In his urine sample, there's basically a huge clonal population can be depicted here. And the most common one is 0, 0, 0, 0, 0, 0. Okay, and there's some 2 side population has one binary call here, all right? 0, 0, 0, 0, 0, 1, or 0, 0, 0, 1, 0, all right? And you look at that. Now, this patient had PSA 5.77, 3 biopsy called GS. It's a high grade. And it's a very interesting thing. This patient has basically binary code that are high grade pins. But if you know, as a pathologist, that basically a lot of patients with prostate cancer actually have a prior history of the precursor lesion, which is shown here. So indeed, this patient could progress from a precursor lesion leading to prostate cancer. How about this patient? Somewhat heterogeneous based on the clonal pattern. But you can see find 4 clonal codes that are been basically highly specific to cancer in addition with the high-grade pins. Okay. Now, this patient has a prostate-specific population of cell in his urine that we can detect. 2 major one, all right? And this is the biopsy result. GS 4+3, 3+4, and 70% of the biopsy core has cancer. But our urine sample can also find a large prostate cancer population likely exists in his urine sample. And look at this is even more dramatic. 57 years old man, PSA 9.5, which is quite high, and 10 out of 10 biopsy core has 80% tumor with GS 3+4, and look at the urine sample has a very basically reduced heterogeneity, and has a large clonal population present. All right, and very interesting thing, this gentleman, 80 years old, look at that, PSA is skyrocket high. Immediately they treat the patients with hormone, anti-hormone therapy, and when we collect urine, after treatment, we cannot find this kind of dramatic examples. Somehow there's only minor population exist. So perhaps this kind of approach, single cell approach, can be utilized for treatment monitoring. So this is a total sample that we study. Basically, you can see normal person has a specific code with a green code that we hopefully can explain that to patient, a couple of years from now, if this can be used for routine non-invasive detection. BPH and high-grade pin. And when a person hit pin, you can see there's basically clonal population exists in your patient's urine sample. And there's a group of prostate cancer confirmed by needle biopsy. They basically look very similar to precursor lesion, okay? Now, this is what we call prostate cancer type 1, prostate cancer type 2 based on urine analysis. This has a lot of clonal populations. And we now know all this has a high risk of prostate cancer with a Gleason score greater than 3+4, okay? Now, a very interesting thing is one of the patient 50, according to the needle biopsy, he actually has a very indolent behavior, but our urine analysis pick up likely he has aggressive cancer. We don't know, all right? And this is potentially a Gleason score upgrading, we don't know. But this can be used to further monitor the patient, whether indeed this patient has indolent cancer or potentially turn into a higher grade cancer. And patient with a highly heterogeneous population still there, okay, usually has a Gleason score 3+3 and smaller tumor. Look at this group, it's pretty similar to normal, except they have more red, all right? So this is a summary of first story is that we are now able to deal with a complex expression system, especially a gene that has a bimodal distribution when you look at the single cell level. In that case, you can convert this to binary code, and with that, then you can build a different type of pattern. Okay, this is what we can handle 2 out of 6. You have 64 code. How about if we look through TCGA cohort now? We are doing that now. And actually, we uncover there's 30-some bimodal genes. Now, can we put 2 out of 30-something? That almost beyond we need a supercomputer to compute different binary code to decipher the complexity of expression pattern at the single cell level. So, the good thing is that we are able to do this in a simple way, and then the end result for this single cell analysis is just a computational algorithm. Okay, so we are now further refined algorithm. The second part of my talk, which I will go quickly, is to look at circulating tumor cell in the blood and realize you can also isolate free nucleic acid. Well, mutation detections. Just a quick background of circulating tumor cell. This is normal or benign patients. And then when you have breast, colorectal, lung mets, or prostate, ovarian, one can see there's sometimes a whole spectrum of circulating tumor cell can be detected in the blood, okay, on average. Now, this is a standard now for patients with prostate cancer. And they have done quite a study. If you, for 7.5 ml of blood, if you find the patient can find 5 single CTC, the survival basically is lower compared a patient has a greater a smaller single cell of CTC. Okay, so this is now, seems to me, it's a standard approach to predict a patient's survival. So the question is, can we do something more with the single cell technology now we have, even though it is extremely rare and we can count number, but can we look at the expression profile and other molecular property and do further predictions? And one of the idea that we have is to look at epithelial to mesenchymal transition profile, or so-called EMT genes. So this is epithelial type, and when they transit to mesenchymal type, the cell shape basically change quite a bit. And during that process, one can have snail, A12, TGF beta twist, CDH2, and Wnt vimentin upregulation and CDH1 downregulations. So can we look at the expression profile by first isolating that single cell in the blood and then look at EMT profile. Right now, the standard approach is using a magnetic bead, okay, or a magnetic system that can identify EpCAM positive CTC. Okay, this is just approach what we call needle in a haystack approach. The other thing is a negative selection by take up all the CD45 positive, they're basically leukocytes, and then enrich a circulating tumor cell. But again, this is a rare cell. We've taken the second approach by look at based on the size. We know now that CTC has a larger size than leukocyte and RBC. By using specific filter or a size filtration methodology, one can retain CTC on a piece of filter or on a piece of platform, and with that, you can use it basically visualize it and use a micromanipulator to do that. So instead of doing the EpCAM, that magnetic bead to take up a group of CTC now, we basically find individual CTC after filter through the filtration device. So you look at here, this is actually individual hole on a piece of filter where the RBC and leukocyte basically can be filtered through. And this is circulating tumor cell. Lo and behold, you can see it, you can visualize it on a piece of filter. Then you can do staining, it's CD45 negative. Then they are not from leukocytes or RBC, and they're EpCAM positive here. Okay, once you identify that, you then prepare the needle. There's two cells here. Just to give you an example, this is the cell of object that are EpCAM positive. Okay, one can just use the needle, go in pinpoint, and take up that single cell, and that this CD45 positive cell still remain in the solutions. Okay, and then one can deposit this cell in a single test tube. And then we subject to microfluidic PCR analysis. All right, so this is the EMT gene panel of patient that the blood that we take that you see there's a PSA doubling. Although the patient is undergoing hormone ablation therapy, so likely cancer could be somewhere. So there's 3 or 4 patients here that we analyze. And this is a patient with castration-resistant but undergoing immunotherapy. And this is a patient that still is sensitive to hormone ablation therapy. One can look at this and see the genes, EMT-related genes are not expressed quite a bit. And this is the summary. Using cumulative gene expression value, one can differentiate these 3 groups. One is castration-sensitive, restricted. The other castration-sensitive. And this is castration-resistant but undergoing immunotherapy, and this is castration-sensitive. So, patient that resists to hormone therapy basically has high EMT value. And that high EMT value translates to activation of Wnt, Sony Hedgehog, and TGF-beta activity. So we are now standardized using this EMT profile for our routine diagnosis. In addition, we began to probe the single cell property of their cell shape. Sorry, I'm just recovered from a cough. We are able to come a very unique technology to poke single cell, all right, and using atomic force microscope. The microscope basically it's been developed to probe the metal to see there's any how soft the metal can be in industrial scale, but we can use that for medical research. And with this AFM that we can probe elasticity, deformity, adhesiveness, and roughness, and that can be characteristic of EMT. So this is the cantilever probe that basically can scan individual cell here and then produce a profile where we can begin to look into the scene, all right? So this is a cell that are stiff. When you're using the probe to touch, you don't see it bounce back, and you don't see it has a more protrusion by the tip. If the cell is more soft, or deformed. And sometimes the cells are very sticky, okay? So when you touch the probe, and then you can to see whether the probe can be removed, or and then that at delay time, one can calculate that. More adhesive here, all right? And this is just a calculation of this kind of characteristic in single circulating tumor cell. So this is just a 2 cell that we take from patient. This patient with a non-castration resistant, castration sensitivity, this is a separate patient. Look at the surface, it's more rough, and the patients with castration resistant, you can see it's not very elastic. And the patient with castration sensitivity, the ball is softer, and this is more sticky, and the cell shape is more deformed. Okay, so we can begin to compare that. This is taken on a total of roughly 250 single circulating tumor cells that we analyzed in this group of patients, okay, in 6 patients with castration sensitive, and then more patients with castration resistant, and you can compare their softness, adhesiveness, and deformation. And one can achieve statistical significance. One can also use in hierarchical clustering to separate them. When you combine the 3 factors together, we can actually separate them quite well. This group of patients are basically castration resistant, and this patient group castration sensitive. So this neuromechanical property together allow us to do this type of analysis. So just to sum up approach that we can do expression analysis of urinary single cell or single circulating tumor cell. We can do physical property. Okay, and then the next step, we are moving to single cell methylation analysis in the pipeline. And we are now also able to do PCR-based protein assay by tagging antibody with PCR-based oligo. And when 2 similar antibodies come together, they hybridize, then the rest is a PCR approach. So this type of new technology, I believe, should be very useful. Today, I only give you an example of urinary exfoliated cell as well as CTC. We now have begun to systematically collect all the needle biopsy ready to be tossed away. Because after all, we don't need a lot of cell. 30 to 50 cells are sufficient enough for us to do analysis. So we can wash the needle and then identify specific cell type by using fluorescent marker. And with that, we can use either using Deb array or fluorescent marker and to do expressions and then the neuromechanical property. We are now in the process of moving to do the targeted sequencing of specific mutation. For example, androgen receptor mutation profile of CTC or urinary cell, and then predict their outcome. All right? So I'd like to thank the lab members who have done this. Joseph and Aaron, and Pei, Susan, and Chun, they are highly trained to do a pig single cell. And then Paul and Maria, they basically the expert of AFM. And Diva and Ian are our clinicians who provide patients' urine and blood sample for us to do single cell analysis. Okay, thank you for your attention, and I can answer some questions.